Everyone's back. Okay, I think we're ready to begin session 2. My name's Kate Baker. I'm a clinical geneticist and a cognitive neuroscientist and a member of the Epilepsy Research UK Scientific Advisory Board. I'm so excited to be here because it just really feels like we're working together as clinicians and as scientists and people with lived experience of epilepsy to focus our attention on the opportunities and the progress that's really going to change lives and is changing lives now. So it really just has a fantastic feeling. This session is about research with people, so building on the amazing molecular and cellular and animal model work from this morning and thinking, how do we bring that into research that involves whole people, whether it's in hospital, home, community settings. And our first speaker is Dr. Rhys Thomas, who will be answering the question, what is clinical research? Dr. Thomas is an honorary consultant in epilepsy at the Royal Victoria Infirmary in Newcastle and a clinical fellow at the university there. Rhys is clinical lead of the UK Epilepsy Priority Setting Partnership. It's a really exciting project that Anna summarised for you. And he is co-director of the Epilepsy Research UK Doctoral Training Centre. So he's really pivotal at training the epilepsy leaders on the clinical side and research side for the future, and I guess being the bridge between your priorities, clinical research. So, Rhys, what is clinical research?
Back. Thank you very much indeed, Kate. And I'm really fortunate to have some of the newly appointed members of that doctoral hub in the audience today, meeting for the first time face-to-face with them. It's really, really exciting. So some of my answer, what is clinical research, was given, I think, in the little video we watched at the beginning. Some of it was actually given by Kate there. And I'm going to talk you through what it means to me, and we'll be able to answer some of the questions then in the panel session afterwards. So, clinical. What does clinical really mean when somebody says that Harry Kane's clinical in front of goal? What does that really mean? Well, clinical comes from a Greek word meaning bed, klinos, so it's at the bedside. And I think that's a really important place to cement us, because here it's a metaphorical bedside, meaning adjacent to the person, adjacent to the patients. I think bedside's a little bit patronising. It's suggesting that the patient's doing nothing to help. There's a laid out flat in front of you, that they're a passive part of research, but that's what clinical research is. You're being observed, you're being watched, you're at the centre of all those kind of surgeons looking down upon you. So what I would say is that clinical research is where you can see the whites of the eyes of the participants or the patients. And so that's the bit I really like. When I worked at McDonald's as a boy, I was put in the kitchens initially. Didn't really like that. I'm not a very good technician. If you asked me to do the same thing repeatedly 10 times, I'd get it wrong 8.5 times out of 10. But if you ask me to be with people, that's the bit I really enjoy. And so the clinical research could be taking information from people, working with people, it could be conversations with people, it could be delivering things back to people. But at all stages, you can see the whites of the eyes of the individuals that you're working with. So what does that really look like? It could be giving something to somebody, which could be a gene therapy. It could be conversational therapy. It could be providing them with lifestyle strategies, making them less likely to go on to have something. So that's delivering something to somebody. Or it could be taking something from them. That might be information that they've given up freely, such as how often they use ambulances. It might be blood samples from which we take DNA. It might be taking something from like a biopsy. So the patient, the teddy bear, the patient is at the centre of clinical research. There's a word that we often use, it's a jargon word. Universities love it, and they talk about translational research. Translation here is moving something from one language to another, so that's moving it from one sphere to another. And it's really tricky and really expensive to do translational research. It's moving information, insights, discoveries from things like my cousin's work in cells and dishes at a basic level, and then moving it back towards the patient at the bedside and hoping that we don't get lost in translation. Because there's lots of really important discoveries that never make it all the way through that pipeline As I said before, because it can be long, dispiriting, expensive, and bureaucratic. That's the translational pipeline. So what do we hope to do with clinical research? Well, I think the nice about clinical research, when you go back to somebody, this is where you really make impact. Just make change, and make change at a human level, a person level. Now that might be that you've improved somebody's treatment. So depending on your worldview, you might look at brain surgery for epilepsy, and you think, well, it certainly helps people in the long term, but by every definition, it's destructive surgery. You're removing something, you're removing tissue. Wouldn't it be more helpful to somebody's brain, their networks, their personality, their emotion, and their memory if we had a treatment that wasn't destructive, like gene therapy? So it's a change in treatment. Or it might be change in policy. How do we go about doing this? This might be health service research. This might be about levelling up. Sorry to use a politically imbued phrase, but it might be best that we stop doing all research for 5 years and actually just benefit in improving the clinical services in areas where there aren't specialist nurses, neurologists, and ambulance people with an interest in epilepsy. What would that do for people with epilepsy in the UK? That's policy research. Or you might think, actually, I don't know, why do people with epilepsy actually, I don't want to steal the thunder of people coming after me. I'll bottle that thought up. Or care. And so that might look like treatment studies. We have these in most major neuroscience units. So this would be people who come from an industry, coming from a pharmaceutical company, and they'll either say, could you help find participants who'll be the first people to ever try these medicines? These would be 5, 10 people, might be healthy volunteers, and we'll see what this medicine does to your body, what you do to the medicine. Or they'll come back at the second stage and say, well, we think it's okay, we think we've got the dose, but could you tell us what the safety profile of this medicine is like? And they're not so interested that stage doesn't work, or they come back at the third stage and they'll say, can you tell us if this medicine works? And we've got people being recruited to those types of studies in every major centre in the UK. Or, as I said, it's policy, letting us know about messaging. You know, how many people in the UK live with epilepsy? What impact does that have on people's lives? What would happen if we reduce that number by 5, 10, 15, 20%? Some of what we're doing sounds as if it's not as exciting as I find it. And that's, for example, just observation, what we call natural history studies. What happens if you just look at the disease trajectory, the condition course? Take, for example, a rare genetic epilepsy. We might know a little bit about what happens between the age of 0 and 5, but many of these people are being diagnosed later and later. What happens to these young adults? Do their seizures behave the same way? Do they live a long, healthy life? Do they age prematurely? These are natural history studies. And for rare disease, this is really important, because if I had a treatment study for a rare disease where there are only 50 people with it living in the UK, am I going to only give the treatment to 25 of those people and have the 25 being untreated? That may not be fair. It may be that we have to use this natural history data as the comparison and give the treatment to everybody that we can find. And clinical research is expensive. Anything that involves combining the NHS and the university is expensive. Anything that involves paying for doctors and nurse time is expensive. Even going as far as finding the dummy pills that you put the medicines is expensive. My wife's a clinical researcher in a parallel field, and she had a treatment that was really only a vitamin, and it came through Pfizer. And they said, well, we'd like to give a dummy version of this vitamin. But Pfizer had copyrighted the colour of the electric pink for this vitamin pill. And this alone meant that the study was kiboshed completely, and they lost their MRC funding. I know. I'll pass on your sympathies to my poor wife. She's long suffering for other reasons as well. The reason why I like clinical research is it's really variable. Working with people is fantastic, it's inspiring, it's humbling, it's difficult, it can be complicated. As you can imagine, trying to bring people to have treatment studies over the last 2 years have been beset by missed appointments and rescheduled things. We've had to be innovative. Can we do more work in people's homes? The answer is yes, of course. Can we do more digital and online observation of people rather than bring people to our centres? The answer is yes, of course. And should we have done this before? Yes, of course, it's also greener. So we can, this is the kind of a wheel of the different types of research. We've got treatment research, which I've mentioned. Prevention research is really exciting and perhaps something that we haven't done enough of within the epilepsy sphere. There's some things that are more obvious. People sometimes have brain injuries and get epilepsy. We do brain injury prevention research, but there'll be some things that are not obvious that we can work with. Screening. We don't often do a great deal of that with an epilepsy. We wait for people to have their seizure and then we are interested in them. But can we start looking for people before they've had their seizure to identify who's at risk of having epilepsy? Diagnostic research, many of which is genetic, and so I won't steal Kate's messaging. Imaging research is absolutely fascinating because we always do scans and tests of people's brains. We harness that information at group level to better understand and predict what's going to happen to people or predict who's going to benefit from brain surgery. Data analysis, one of the benefits of epilepsy being a common condition. There are so many of us working with this epilepsy community. We can harness this information from paramedic right the way through to brain surgery to work out new insights in terms of people and their seizures, and then health services research that I touched on before, which is really important and underfunded. So I would like to say that the best clinical researchers are a bit of a Swiss Army knife. They're adaptable. They can turn themselves to lots of different situations. And this is me being on the front foot and using this as a metaphor rather than saying jack of all trades, master of none. Because if you're somebody who's going to pivot from health services research to communication research to priorities research, you've got to be able to have many clubs in your bag, lots of tools at your disposal. The patient experience so far has been missing. I've been talking about what's been researcher-driven, researcher-led. So let's talk about what does clinical research look like to the individual. I think it matters in terms of where this happens. The vast majority is going to be outpatient research, that's people giving you medicines, you go home with those medicines and you fill in questionnaires and you come back 10 times in 12 weeks to see how you're getting on. Inpatient research is really important, and often we use the fact that people come in for EEG monitoring for this inpatient opportunity, or we tack on additional research questions when people are having things like brain surgery, things that are very intense. There are things that are intervention, means that we're changing something about your experience or your treatment. Those come with a lot more ethical red tape, a lot more forms than observational research, where you may not necessarily know that you've been a participant, that you've been helping in observation research. So, for example, in my hospital in Newcastle, just by stepping in the front door, it's a university hospital, you've given yourself permission, given us permission for us to look at your health record data at an anonymous level. Whether you believe it's correct or not, I can look at people that I don't look after. So I can look at people, anybody with seizures, anybody with a health condition using this portal, this kind of observation data. But I couldn't use that information to go back to you. I couldn't change anything with that. Then you've got registries, really good for rare conditions. So if Mike suddenly realizes that he's got a drug that he's screened through the cells in a dish that doesn't just help people with DNM1, but might help sister and brother conditions, might go to other people who hold registries of rare disease across the world saying, could you try this in these conditions as well? So you can rapidly upscale some of those insights. That's still using the idea of the patient as a participant in research. And one of the things that we are really keen to do and change in terms of the profile is the patient as a partner. There's a lot of lip service paid to this. It doesn't always work as nicely in the real world as people would like to have it believe, but there are some good examples of research questions that come directly from individuals. People ask you a killer question in clinic, and ask, why can't we do this? And you stop and you think, oh, I wish that was my question. Oh, that's great. That's great. And if you come up to me in the break, I'll give you the example of the most recent one of those that I've not had a good answer to yet. So I think it's a killer question. So one of the better examples is trying to make sure that all of the research that we're funding, that we're prioritizing, and all the work that we are sort of putting our limited resources in towards is important. Now, you might say, I think it's really important to know that what's the best type of exercise to prevent yourself having seizures. But if I only gave you a limited budget, is that the one question that you want me to fund? Is that the one most important one? So if you had a limited set of money, set of researchers, set of time, what would we focus upon? Now, Gareth said that epilepsy is a common condition. From first day to your last. And so we've got a huge experience we need to draw upon. So it's been great that Anna showed us that we've been overwhelmed by people giving us their research priorities for this epilepsy priority setting partnership. We've got to be one of the largest. It's got to be either us or bipolar. It's got to be one of the, we've got to be one of the largest. So our community has absolutely come out in spades to help us here. And we're on time to be publishing this in the autumn, which is really exciting, where we can give you a top 10. You may not agree with them, but you can always see where the questions have come from. And those questions will generate really important, prioritised areas of research for us in the UK going forwards. So that's the patient as a partner. Clinical work, clinical research work, hard work, expensive, but really richly rewarding. And there are still people that I remember meeting doing my PhD that would come to a dingy room and sit doing psychological testing, and I'd take their blood and look at scans and things like that that I'm still in contact with today. So I'm going to stop it there and we'll take the questions at the end. Thank you very much. So next up we have Dr. Kate Baker. So Kate's going to be answering another tricky question. Can genetic research help solve unanswered questions for epilepsy? Now, normally when the question is the title of the question, the answer is no, but I strongly feel this is going to be a yes. I strongly feel it's going to be a yes. So Dr. Baker is a program track leader at the MRC Cognition and Brain Sciences unit at the University of Cambridge and leads the Genetic Disorders and Cognitive Development program. Kate is an honorary consultant in clinical genetics at Cambridge University Hospital. Podium's yours.
Thanks so much. We're really lucky to do the work that we do. We really enjoy it as well. So, can genetics research solve unanswered questions for epilepsy? So I was going to start with a vote, actually. So, broadly speaking, who thinks the answer is no? Yeah, and broadly speaking, who thinks the answer is yes? Great. Okay. So let's work it out together. A bit of a reminder, just to make sure we're all on the same track to answer this question together. So epilepsy and genetics, what's the connection? Mike this morning reminded us that we're built of cells, and at the centre of every cell is the nucleus, and the nucleus contains all the genetic information that your cells need to grow and to survive. And that genetic information is packaged up into these things called chromosomes. Every cell contains 23 pairs of chromosomes, one of each pair from your dad, one of each pair from your mum. And chromosomes are built of this chemical called DNA. A stretch of DNA is a gene, and a gene is an instruction to build a protein, usually. So what are proteins? And we're used to sort of eating protein, but proteins are the molecular building blocks of the cell that inform the shape of the cell, the biochemical functions of the cell, and the way that the cells interact to become a tissue, a part of the body. So proteins are your kind of toolkit inside each cell. Why are proteins important for your brain? Because your brain is this complex network of brain cells. And so the proteins inside your brain cells are what regulate your brain cells' structure and function. What's the connection to epilepsy? Well, you can have a very, very tiny difference, just one change in a sequence of DNA for a gene. It's unbelievable, really, the importance of that. All of our DNA is different. Our DNA variation is what informs our uniqueness as human beings. And most DNA variation is just tolerated. It's either completely neutral, has no effect on proteins and on brain function, or it is part of what informs our characteristics as unique human beings. You can have just one tiny change in a base pair of DNA that can have a drastic impact on people's brain health. Sometimes it's a sequence change. Sometimes, as we heard earlier, it may be a missing chunk of DNA, a deletion or a duplication, or a change in the shape of a chromosome. But the impact of that is the change in a protein that's important for your brain, either the amount that that protein is made, or its shape, or its aspect of its function. So one tiny DNA difference can make all the difference to a key protein in your brain. And that lightning bolt is it can change the likelihood of developing epilepsy. It can never say exactly when someone's going to have a seizure, or perhaps what type of seizure they're going to have, or how they're going to respond to treatment. But it can be the key defining cause of somebody's epilepsy. So DNA differences are one of the main underlying causes of epilepsy. That's the connection. And here are some facts about epilepsy and genetics here and now. So there has been a technological revolution in our ability to gain knowledge about individuals' genetics. And that's driven by this broad term next generation sequencing. And here and now, there's up to a 50% chance of finding the genetic cause of a person's epilepsy. That's radically different from 10 years or 20 years. This is a new window through which to understand epilepsy that simply wasn't here before. And this complex graph from a lovely summary paper just shows you time along the bottom from 1991 to only 2017, and now we're a few years on from that, and the trajectories continue to grow. And up the side of the graph, as someone mentioned earlier, I think Alison mentioned, that we now know of more than 500 single specific genetic disorders that can cause epilepsy in some or all people with that genetic disorder. This is completely new knowledge, a new window of opportunity that wasn't here before. Now, I've said up to 50% chance because the chance of finding a genetic diagnosis is not the same for every person with epilepsy. The chance is highest if the epilepsy started early in life. So highest in early childhood, highest under the age of 2. That doesn't mean everybody under the age of 2 is going to have a genetic cause, and it certainly doesn't mean that somebody whose seizures didn't start until they were 15 or 30. It's just that the chance is highest. It's also highest if it's an epilepsy that's difficult to manage with current medications, and it's highest if epilepsy doesn't occur on its own, but as part of a broader picture. So if somebody is born with physical problems that are present at birth, like a heart problem or a palate problem or an immune problem that's present early in life, that means the chance of finding a genetic diagnosis is higher. And the chance of finding a diagnosis is higher if epilepsy is associated with learning difficulties as well. That doesn't mean that people with epilepsy on its own, as their primary symptom, don't have a genetic diagnosis. It just shifts the likelihood. And those likelihoods shift what type of genetic testing will be offered to somebody and what's the chance of finding their clear genetic cause. We now have a reorganised NHS in England genomics medicine service. We've always had clinical genetics, it's not new. There's always been regional genetic centres, but it's been really quite amazingly reorganised in the last couple of years. So we have a national genomic test directory. That's important because that means whether you're in Stockport or Stevenage, the same clinical criteria should be used to guide whether you're offered genetic testing and what type of genetic testing you had. So it should be about removing that postcode lottery. It also means that technology that's applied to genetic testing and the pipelines for interpreting results are centralised. That should be fairer and it should be more efficient. It should become quicker. It isn't at the moment, but it should be. And we still have our regional genetics centres, which are really important because my kind of genetics isn't really looking at sequence. My kind of genetics is having conversations with people. Genetics is about what's your life story? Where do you think your epilepsies come from? What do you feel about passing epilepsy risk on to members of your family? How do you want to use this information to maximise your quality of life for the future and for your family. And those aren't technological questions, those are human questions. So the hope is it works like this. Somebody with epilepsy comes into clinic with their genome and rapidly obtains the clear-cut genetic diagnosis, which opens the post-diagnostic door to you. And once you walk through that post-diagnostic door of knowing the cause of your epilepsy, you have a really clear explanation from the genetics, through the cells, through the systems, to your individual symptoms. You have very accurate information about what's the chance of somebody else in my family developing epilepsy or other problems, be that your siblings, your own children, or other wider members of the family. And you have good, useful information about prognosis. And that's a medical word which basically means what's going to happen in the future. What's that landscape? What's that range of possibilities for my epilepsy, my cognitive development, for mental health and physical health? And as we've already heard really excitingly this morning, that diagnosis turns the key towards personalised treatment that understands your symptoms, the cause of your epilepsy and the mechanisms linking the two, and can impact on your seizures and other aspects of quality of life. That's the hope. And to some degree, there's a lot of hype in that, because is it like that now? And some of you, I read somebody, met someone in coffee who's been through this process quite recently with their family, and probably is standing looking through this door and seeing what does the future look like. It's not quite that sunny. It can feel like we don't have the information we really want to translate genetic diagnosis into better quality of care and a better future for families. And it can feel like actually kind of wish I'd shut the door and just stayed with what I knew before. But we're starting to gather that information slowly but surely. It takes a lot of different sorts of research to build on genetic diagnosis and provide better post-diagnostic answers for more families. And I'm really excited about the fact that we are moving along this journey with a lot of different disorders in ways that we just couldn't do 5 or 10 years ago. But it's not easy, and it doesn't stop there. The answer is more research. So what kind of research do we need? I don't know how I'm doing with time, but what I'm really thinking about at the moment is that there are very different kinds of clinical research questions that arise depending upon when in somebody's life journey they're receiving their genetic diagnosis, the patient themselves and their family. So sometimes genetic testing is carried out before someone's born for a variety of reasons. Sometimes when they're very first born, perhaps developing seizures in the first few days or weeks of life and they're in the neonatal intensive care unit. Sometimes during early years when they're at nursery and then develop seizures, and perhaps there's lots of concerns about their development. Sometimes we don't do genetic testing until later in childhood. Problems may have been there for a while, but now we may have a new genetic diagnostic answer, or emerging seizures and emerging learning difficulties and other problems. Then we often offer testing now in adolescence, because there's a whole generation of children and young people growing up who have probably the most to benefit from new genetic diagnostics, and we need to catch up and open this door for a better future for this generation currently growing up. Adults are people that amazingly grow up, and when I gave this talk to some of the lab recently where I work and said, well, we need to be prioritising genetic testing for epilepsy in adults, they said, well, we don't have a panel for that. I said, no, look, there's a whole 2 generations of individuals whose epilepsy started before the age of 2, and they have these conditions we're now diagnosing, and they have a host of physical medical problems and mental health difficulties and concerns about their older life care. They exist, and it was a kind of light bulb moment. The lab, they're very intelligent people, but they're so focused on getting in diagnostics as early as possible, they're forgetting that actually we have a duty of care to the existing patient group and a huge amount to learn from those generations as well. So the kinds of questions that arise in clinic early on are often about survival and severity and prognosis and early treatment. And is there a window of opportunity for changing the future if we do diagnosis in the early years of life and disease-modifying treatments that could alter the cognitive course of someone's condition? Those are the early questions. But in the early years of childhood, adolescence, which is actually the research I do, the priorities are communication, learning, relationships, social inclusion, managing challenging behaviour, living skills, teenagers and young adults' own relationships, recurrence for the next generation, and in later adulthood, promoting physical health, reducing the inequalities in length of life and in other aspects of physical health morbidity. There are different priorities, and it takes a lot of different sorts of research. So when people think about genetics research, I think about sequencing, that's really important. Better accuracy, reducing variants of uncertain significance, very important. Experimental models we heard this morning, very important. Cohorts, longitudinal stuff, developmental psychology and cognitive neuroscience, that's my patch. How do we do clinical trials for rare conditions? And then how do we implement that into better integrated models of holistic care for patients and families? And some of my research is about family research. What's the impact on parent-child relationships of making a genetic diagnosis, for example? And parenting care and mental health is something we're focusing on very much. So the last 30 seconds of what I'm doing, I haven't got time to tell you examples of any actual studies, but what we're trying to do is move closer towards this brighter future by gathering a lot of that natural history information Reece talked about, and actually focusing on the disorders Mike talked about. Let's think about groups of epilepsies that arise out of similar aspects of cellular dysfunction. Let's map the range of different differences in vision, in movement, in emotion control, in attention, in impulse regulation. Let's say, how is that group similar, but critically, how variable are they? How are they different? Because those of you who may have been through a genetic diagnosis process know, well, there's a person with a genetic diagnosis, you look at the literature and you go, well, yes, some of that fits, and some of it really doesn't, because no two people with the same genetic diagnosis are the same as each other. So we need to understand variability as much as similarity, and that's the kind of work that I do with a little team of researchers, and we love going out around the UK, coming to meet you at home, sitting at a kitchen table, and just basically understanding the life stories of people with these rare genetic disorders, and then designing our cognitive hypotheses about, well, where and how is brain development different depending upon genetic cause, and building those bridges between the genes and the cells and the everyday experiences of particularly the cognitive and mental health side. And as I mentioned, I also have a line of research on families of saying, well, there's always been a focus and a line of research on the mental health needs of particularly parents caring for individuals with long-standing conditions. I'm just asking a new question. Well, what difference does genetic diagnosis make to parents and their mental health? Does it matter when that diagnosis is made? Does it matter what type of genetic diagnosis is made? How long does that genetic diagnostic journey impact on people's anxiety levels out in the real world? Because I don't have all the answers, but I think these are really important questions to ask, because people with epilepsy don't live in boxes. They live as part of families and communities, and our research needs to address those needs as well. Thank you. It's great to have an opportunity to share some thoughts, but what I really want to know is, with epilepsy and genetics, what do you think the questions should be? Thanks. Great. And I believe that we have Professor Roiber joining us online, who is a digital doctor. So I feel like saying, Dr. Roiber, can you hear us? Aha, there he is. So Marcus Roiber is another member of the ERUK SAC, Scientific Advisory Committee, and he's a professor of clinical neurology at the University of Sheffield and an honorary consultant neurologist at the Sheffield Teaching Hospitals NHS Foundation Trust. And he is editor-in-chief of Seizure, the European Journal of Epilepsy. So those of you who don't work in research may not know just how powerful and important journal editors are, because our research is only of any value if we're able to communicate it to other scientists, and he's in charge of that bit. So Professor Roiber is going to tell us about digital doctors, technology in epilepsy clinics.
Yes, thank you very much for providing me with the opportunity to talk to you. And I must apologize that I'm not there in person. I do exist in person, so I'm not just digital. But unfortunately, I couldn't come to London today. So yes, a brief run through technology in the lives of people with epilepsy today and in the future. So I'm going to go through the process of being diagnosed and treated with epilepsy and living with epilepsy. So starting with the diagnosis, this today, of course, is a very low-tech procedure. You ideally talk to a doctor, maybe face-to-face or increasingly over the telephone, and you have the doctor, an expert, listening to that story and deciding whether it is most likely that you have epilepsy or perhaps one of the other common causes of transient loss of consciousness, such as fainting or dissociative seizures, so a response of the body to some kind of stressful trigger. There is a lot more tech involved in investigating people with epilepsy. However, these investigations, even the most fancy ones, on the whole can't tell us for certain whether you've got epilepsy or syncope or dissociative seizures, so they can't help with the first question. That would only be possible in a relatively small number of people in whom we can record their attacks, but this would only be relevant for people who have quite frequent attacks. So in most cases, technology doesn't help with the initial diagnostic question. So technology is then reserved for investigating what kind of epilepsy it might be and what the most best treatment might be. And we have very fancy tests, especially when we get to image patients where we're thinking about epilepsy surgery, aiming to remove a part of the brain. So for instance, we do tractography, that's the very colorful images at the top there that show us about the wiring in the brain. We can do PET scans, that's bottom left, which show us which parts of the brain use most sugar, which parts are most active in between their seizures. In addition to different MRI scanning techniques, like the top left that maybe you're familiar with, we can also image the magnetic field around the brain to show us where epileptic activity is arising. But all these tests are really relevant only for a small minority of patients, and increasingly the technology involved in producing images has moved faster than our ability to interpret these images optimally. When it comes to treatment, what we do in terms of tech at the moment is still limited, but there is scope for improvement. So a standard treatment available in the NHS is depicted on the right. This is vagus nerve stimulation. This involves an electrical device that is implanted under the skin of the left shoulder, and a wire that is also running under the skin and attached to the vagus nerve. This is a nerve that travels from the brain to the body through the neck. The electrical device sends electrical impulses through the vagus nerve into the brain and is intended to stop electrical activity from building up to the sort of pattern that will cause an epileptic seizure. The devices can come on automatically every few minutes, but they can also monitor your heart rate. And if the device senses that the heart rate is suddenly increased in a way that it might during an epileptic seizure, the device could also begin to fire at that moment to try and disrupt the seizure. So that's the latest technology that's currently available in the NHS. On the left, you see a stimulation system that is implanted directly into the brain. This is also commercially available, but currently not in use in the UK. So this is a treatment available, for instance, in the States. So with the most advanced form of this treatment, you've got a device implanted in the skull with a wire that is placed in the brain and which can read the EEG and also deliver electrical impulses directly to the brain. So in some ways it's similar to the vagus nerve stimulator, but we can target different areas in the brain with this device. Both of these treatments currently don't really stop seizures altogether. They reduce the number of seizures by over 50%, maybe in 2/3 of individuals who receive these treatments. So they work, but there is still some scope for improvement. When it comes to living with seizures, on the whole, our approach is fairly low tech. So those individuals who keep seizure diaries often use paper and pencil diaries, or even if they use electronic diaries, then they rely on their ability to remember that they've had a seizure, or on people around them who remind them that they've had a seizure. But we know from research studies that 1/3 of seizures are unreported, for instance, because they've happened in sleep, or because the person who's had the seizure is not in a state immediately after the seizure to fill in a seizure diary. And then when they have recovered, they've no recollection of the seizure having happened. or a person has no subjective knowledge of the seizure at any point. So these sorts of seizure recordings are suboptimal and can be a real problem if seizures only ever occur at night and you never know whether or not a person's become seizure-free with a form of treatment. When it comes to trying to remember tablets, we use tablet boxes like the one depicted here. When it comes to warning, giving people warning of seizures, technology is even older fashioned. Some of us, some patients have seizure dogs that alert them, either trained ones or their own homebred dog has developed a sense of a seizure warning. We do use seizure matrices in some individuals to detect tonic-clonic seizures in sleep, but of course these are also quite limited and only work for major seizures. And on the whole, just tell you that the seizure has just happened, although they can sometimes alert people who might want to help. So how might the future look with regard to technology? Well, at the moment, we're heavily reliant on experts, and there are not enough experts around when it comes to making a diagnosis. What is more, the diagnostic process is quite crude. So we just say, oh, I think this is epilepsy, or I'm very sure this is epilepsy. So we have some kind of idea of likelihood, but we can't put a number to that idea, how likely it is, or how much more likely it is that one person epilepsy than another. So we've been working on a digital system where people can talk to a digital doctor presented on a computer screen, answer questions. And that system worked like it's depicted on the right. So you have speech, which is put into the computer and subjected to something called automatic speech recognition, which turns your spoken words into a typed script. You can do diarization, which would split words spoken by a patient with epilepsy, for instance, from a witness. And then you program the computer to look for certain diagnostic features in the transcript that you have produced. And then you use something called a classifier. That's another computer program based on these features to decide whether the computer thinks it's more likely that you've got talk of a person with epilepsy or talk with a person with another condition. Epilepsy Research UK has funded some further research with this system. For instance, I'm just going to show you two studies like this. So in this study, we looked for signs of formulation effort, people trying very hard to communicate something that is really difficult to explain. We know from previous research that people with epilepsy find it particularly difficult to explain their seizure experiences. So we got the computer automatically look for hesitations, for repetitions in speech, and for any expressions of uncertainty, like it's kind of, or it's sort of, or I don't really know, those sorts of expressions. We also measured a number of different types of pauses. And with these automatic features, we were able to classify over 2/3 of people correctly. So this was a computer who knew nothing about epilepsy, and all the computer had to decide on was the transcript of speech. In this further study, we used a different method. So in this method, in this study, we used something called linguistic inquiry and word counter applications. So that people have programmed all sorts of word clouds that relate to particular observations like emotions or particular kind of emotions, or also persons, groups. So we took some of these predefined categories and ran them over our transcripts. And with this method, we were able to classify 80% of patients correctly into those with epilepsy and those with other causes of attacks. So this may sound crazy, but I think it is not impossible that at one point in the future, a digital doctor can at least give some direction to investigation and produce a probability of a particular diagnosis, even if they don't make the final diagnosis. We can, of course, combine these sorts of speech analysis with a cleverer analysis of the symptoms that people have. So this is from a study where we asked 100 people with syncope, 100 people with epilepsy, 100 people with dissociative seizures, whether they had one of 87 symptoms. And you can see that the profiles of these symptoms are quite different in the three different diagnostic groups, which explain most cases of transient loss of consciousness. So I think we can use data that is collected with questionnaires, for instance, or digital questionnaires, to produce precise diagnostic predictions, which can help us interpret these clever investigations that I mentioned earlier, like the MRI scans that show possible abnormalities, or maybe they're normal, or maybe it is meaningful, or maybe it is not meaningful. In terms of stimulator technology, there's a lot more that we can learn about different types of the brain, different areas of the brain that we can stimulate, different ways of stimulating. We already have access to non-invasive vagus nerve stimulators that people can put in their ears, for instance, for a quarter of an hour in the evening, or half an hour in the evening, to achieve a measurable effect. What we don't know yet is whether these non-invasive means of stimulating the vagus nerve or the trigeminal nerve, or applying something called direct current to the brain, whether these techniques are as good as the vagus nerve stimulator technique, which involves a big operation, or the brain stimulator technique that I told you earlier. I think the biggest and most imminent change in technology is what's going to happen to living with seizures. So many of us are now using smartwatches, which monitor our fitness, and we record our heart rate as we walk about. We do step counts and so forth. Well, actually, this sort of seizure watches have become quite good at identifying tonic-clonic seizures, and they're getting better at identifying other seizures. So these are devices that could, for a start, automatically and reliably count seizures, better perhaps than a diary. And also devices that can help with seizure forecasting, that can identify periods when we're at particular risk of seizures. There are smart pills and smart bottles. The smart bottles can send a message to your phone, or can check with your phone that you have already opened the bottle at round about the right time of day. The smart pills can include technology in a tablet that sends a message to your iPhone from your stomach that the tablet has actually arrived in the stomach. So these technologies are available. They can help to remind people whether they've taken the tablets. Of course, this information could also be passed on to your care team to inform them whether you have taken the medication as prescribed. So, dashboards have been designed that would provide information to patients, but also to their treatment team, their clinicians, about seizures they've had, tablets they've taken, and their level of activity. So I think there's a lot of technology out there. There's much more we need to learn about it to know that it functions well in people with epilepsy. And there are also questions that you might need to answer about how much transparency you want, how much information you want to share with your doctor, because there's a whole lot of information that you could now share with technology that is available. So thank you very much for listening. This, by the way, is Nathan, the guy who's done the research I've shown you.